Okay, so now we've got prophase taken care of. Oh, no, no, no. What have I done? Okay. Prophase taken care of. And I can show you, it's really hard to see these, but here are where the spindle fibers start to come out. All right, put your pencil down for a minute. Do this. And I know we've all seen this movie. Give me your spirit fingers. You've seen the what's it called? Bring It On? Yeah, Bring It On maybe? Got your spirit fingers. Come on, boys. Okay, your spirit fingers are your spirit fingers, SF. Spirit fingers are spindle fibers, okay? They shoot out, and they're going to grab the chromosomes from the middle, okay? So if you don't know what spirit fingers are, it's the 21st century. Go watch that movie. Okay, metaphase. Metaphase is your next phase. Here's how I would draw this if I were drawing a plant. I would draw my cell wall that's still outside. I would draw my spindle fibers shooting out, but now they're actually touching, they're actually touching the chromosomes. Okay, draw yourself, your spindle fibers touching the chromosomes. Tell me if I'm going too fast. But where are they touching the chromosomes? What's that middle thing called? Centromere. Yes, there's a dot here in the middle. Centromere, kind of sounds the same. Center of sister chromatids. The spindle fiber is attached to the center of the sister chromatids. Yeah, Isaiah.
Spindle fibers stem from the central?
In the plant cell, they actually stem from the cell wall. That's a really, really good question, actually. In a plant cell, they come from the cell wall. It just kind of shoots out the rest of the proteins. In an animal cell, they come from the centriole. So I'm actually going to write that right here because I need to mention that later, but I'll mention it now. So spindle fibers from centrioles in animals. Okay, that's the thing that helped with cell division back when we did cells. Okay, and it says the chromosomes go to the equator of the cell. I just remember meta, middle. Meta, middle. That's the middle of the cell. Those are my little hints on how you can remember these things. Give me a thumbs up when you're good. You got your cell drawn, you got your notes taken for. Yeah.
So for the animal cell, we just draw the spindle fibers attaching to the centromere just in a circle.
Yeah, I would draw like, here's my animal cell. The membrane is gone, right?
Yeah,
the dotted line's gone.
Fully gone.
And then you got your little, I'm going to draw this. These two things right here are my centrioles. And then I've got my X's in the middle. And they attach like this.
Thank you.
Yes, yes. Alright, thumbs up when you're good. Not many of us. Okay, some of us are still drawing that. I'll give you like 30 more seconds there. If you're waiting on me, I will tell you something to put in the interphase part. Interphase. Chromatin looks like blob. If you're trying to wait, if you're waiting on the rest of the crew here. Interphase. Chromatin looks like blob. That's important. I'll tell you more later. Alright, told you this is fast and furious. You're fairly familiar with this mess. Next, we have, what's the next phase? Anaphase. Okay, anaphase. The centromeres split. Who's pulling them apart? Thank you. By spindle fibers. They're split by the spindle fibers. They rip those babies apart. And the sister chromatids start to move to opposite ends, so they're still moving to opposite ends. They're not all the way there yet. The way I remember this is that I remember anaphase, away. They move away from one another. So if I was drawing a plant cell, you can see my spindle fibers are attached to my chromosomes on these sides. And the reason it looks like a little V is because it's supposed to be like representing it's like really fast. And if this is my sister chromatid, it goes like that. That's why it looks like a V. You see that it's not just because it's a V. It's just it's like flying across the cell. Okay, and then in this case, I only have half of the chromosome because the other half's on the other side. You can see the blue ones. If I was drawing an animal cell, I would draw a circle with my spindle fibers. And then I would draw my chromosomes like this. That have been ripped apart. What's that? This right here, it's starting to form something called a cell plate, but here's where you really see it. So I'm not mentioning it here, I want to mention it here. Okay? Now telophase, that prefix means something. What do you look at the stars with? A telescope. What do you look at? Okay, well, let's stick with telescope. Telescope is going to be looking at things that are really, really, really far away, okay? So the word tele here, telephase, one, tele, two, it makes two cells, and tele, like telescope, means they are far, 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 far away. That's why they're on totally opposite ends of the cells. They're really far away from each other. Opposite ends of this cell. So this is two distinct nuclei start to form. And what do you see happening around the chromosomes again? Right, what is this? Right, the nuclear envelope is forming again. That's why it says nuclei. So make sure you know the nuclear envelope or membrane, whatever you want to call it, forms again. Spindle fibers, I mean, they're still kind of there, but they've pretty much exhausted their job. They're done. They've ripped the chromosomes apart. But special things happen in telophase between two different cells. This is a plant cell, yes? Plant cells form this division called a cell plate. Because they don't, I mean, they start to split their cytoplasm, but they start to form the cell plate now, but they finish it in cytokinesis. So cell plate starts to form. That is very, very, very important. So even if you're not drawing a plant cell, I need you to know in plant cells, a cell plate forms. So if I asked you, what phase does a cell plate start to form in a plant cell? You'd say telophase. Thank you. A couple of us are still drawing there. If you've drawn an animal cell, it might be to your benefit just for the telophase one to draw this plant one. Can I scroll up? Is that okay? Telophase right there. Now I'm going to show you what the animal cell looks like. I'll make sure I don't have it on that slide already. Okay. Okay, an animal cell was what I was talking about. We know the animal cell has this circular appearance. Well, it starts to form this like little dip. Okay, it starts to pinch in. You can see it pinching in here and here, yes? It's not all the way pinched in and connected, but it starts to. So this is my animal cell. Let's see here. Da 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 da. How many chromosomes in each cell? 4. My nuclear envelope is starting to form again around them. This pinching thing is called a cleavage. Did I spell cleavage right? I got it. Spelling in capital letters. I don't spell capital letters very well. Cleavage furrow. Okay, it starts in telophase but finishes in cytokinesis just like the cell plate did. Cleavage. We all know what cleavage is. Just like we all knew what the other one was when we talked about stomates. The reason this we call it cleavage is because it starts to look like this. Yeah? You get my drift? Okay. I'm pretty sure the cleavage furrow word came before the other one did. But cleavage furrow is what we call it for animal cells, and that makes sense because only animals can have cleavage. Plants can have cleavage, right?
Oh.
No, plants can have cleavage. All right, so let's actually see what that looks like, okay? Looking at this one, you see this big bundle? I have no idea how many chromosomes are there. Which phase is that? Well, I can see that they're condensed already. They have condensed because you can see, like, there's a chromosome, there's a chromosome. Huh? That is actually prophase, yes. That is prophase because you can see the condensed versions. It's just, it's a mess, but it's not like thin lines, the thin spaghetti. That would be chromatin. That would be interphase. All right, we can see they're in the middle. Metaphase. We can see they've split. Anaphase, we can see two distinct nuclei. Two distinct nuclei, two telo, right? And then you can kind of see it's starting to form this like pinching motion here. So it's starting to form the what? Cleavage furrow. Okay, now let's finish it up. We got cytokinesis to take care of. Right here. Cytokinesis has a lot of information. Write down everything there because the verbs, like the verbiage, the daughter cells, all that stuff, you got to know specifics here. And I don't have my watch today. So one important part, cytoplasm splits, cytokinesis, cytoplasm. These are some beautiful pictures. And for this final picture, what I would draw is the total separation of the cells. So if I was a plant, the total separation looks like this. Nucleus, 4 chromosomes. Nucleus, 4 chromosomes. What's that thing called?
So,
Plate, right. Cell plate right here. It's very dark this time. It's like saying, okay, now I've got 2 cells. Because plants don't have to make, like, they don't have to literally separate. They just have to make a division between them. Because plant cells build things a lot better than animal cells do. If I was an animal cell, I would have 2 separate nuclei, or 2 separate cells in the nucleus. How many chromosomes in each nucleus here? 4. And the cleavage furrow has finished. Finished separating everything. Yeah. Yeah. The main step here, though, is that the cytoplasm separates. Now, it says daughter cells that are identical to the parent cell. So if I was saying, okay, guys, looking at these pictures, how many chromosomes does the parent cell have? You would say 4, because it's identical. Identical means the same. So in this case, there are 4 chromosomes in the parent cell, and in each of these daughter cells, there are 4 chromosomes. If my daughter cells had 112 chromosomes, how many did the parent have? 112, right? It's identical. They have to be the same. So at the very, very, very beginning, at the very, very end, it goes back to the very, very beginning, which is interphase again. I didn't draw one like that yet for you. I had a couple of you guys write down, it's a blob, so I'm going to draw that here. So interphase, you have your cell, you have your nuclear membrane still intact, but it's a blob. It's just like spaghetti everywhere, okay? And you already have kind of this drawn on your other paper, your cell plate paper. But I want you to know that originally it starts off as a single chromosome, but then during S phase, what happens during S phase?
They start to unravel.
What does S mean? Synthesis. So what's it doing to the chromosomes? Synthesizing another one, right? So there's the one, and then synthesizing it means it makes another one. And what's it connected with? Centromere, right. So it starts off as a unreplicated, and it goes to, during synthesis, a replicated one. And that's happening during interphase. That's why I drew that there. Alrighty. I have one more. I'm going to see that video play, and I have one more thing to show you here. And I'll let you guys work. I'll leave it there for just a second. Questions? I will in just a minute, okay? I think some people are finishing this one. This is all happening in cytokinesis. Or this is happening in interphase, yes. I have 2 questions for you, then we'll move on. Where did centrioles appear? In which types of cells?
Animals.
Animal cells only. Centrioles, you have to know that. Centrioles, animal cells only. Centrioles, animal cells only. Got it?
Yeah.
Star that. Write it someplace so you know. Yes, ma'am. All right, that's one question. Second question. Why in the world will we be doing mitosis? What is the purpose of mitosis? Alex?
To grow.
To grow?
To expand.
Okay. What else?
To replace damaged cells.
Okay. Anything else?
Rebuild.
Rebuild broken or dead cells. What about this one? Asexual is mitosis, yes or no?
Yes.
Yes, please note, and someplace write this on your paper. These are the three functions, the three purposes, or functions, purposes of mitosis. Growth, making more of your own cells. Repair, mending something you've dumbly done, like break your ankle or something. It's an accident, right? Nobody purposely falls down the stairs or gets hurt in basketball and blood everywhere. And then asexual reproduction. Specifically, examples would be like single-celled organisms. Single-celled organisms do this one. Because that's how they make babies. That's how they make more themselves. They have to do it via mitosis. And I have a proposition for you. You're going to identify these. Oh, actually, they're already identified, but I'm going to show you some. You're going to identify these, and then I'm going to ask you guys if you will do a dance with me. Not the photosynthesis dance. That was cool, but this one's way cooler because we might get to go outside for it. Okay, so let's get through this. I have some pictures. My pictures are. Ooh, what is that? Ew. It's not a wart. That's it. What kind of cell? How do you know it's animal? You can see the membrane here. How do you know this is a plant? Look at this flare, and it's green. It's not a nipple. I already heard that. It is a interphase part of the cell. This one? Okay, you see, look at the little bitty, kind of like chromosomes. They're darkened. They're supposed to be X's, but you just can't really see that very well. Okay, but you can still see the nucleus, right? You can still see that membrane. Okay, continue. Can you see the membrane anymore? No, the membrane is gone. What are these things shooting out the sides? Spindle fibers. This one, you can't see them that well, but they're there. All right, how do you know it's metaphase? Middle. Okay. What's happening? Oh, no! We're breaking apart. We're going away from each other. Anaphase, away. And then, what's starting to happen, though? Okay, so you can see the cell membrane has this little cleavage furrow that's starting to form here. And then this one's starting to form a cell plate, because that's what plants do. Plate, plant, okay? Plate, plant. And then cytokinesis is happening kind of at the same time. I don't have a separate picture for cytokinesis. Okay. Let me show you one more thing. Everybody, put your hands together. Okay, get your hands together. Now, what does a blob look like? A blob. A blob looks like this. You can't see any fingers, no chromosomes, no nothing. Okay, it's a blob. Prophase, the chromosomes, they condense and they start to come into appearance. So they go like this. Do this with me. Okay? They start to come into appearance, but you can't really see the chromosomes all the way yet. But they start to come into appearance. What happens in metaphase? Middle. Okay, they're all lined up in the middle. Anaphase? Pull apart. Telophase? Two separate cells. Cytokinesis? Okay, again. Interphase. Prophase. Anaphase. Telophase. Cytokinesis. You gotta get a mean face like. Okay, one more time. Interphase. Prophase. Metaphase. Anaphase. Telophase. Cytokinesis. Okay, here's what I want to do. I want to simulate mitosis in like 5 minutes outside. However, you don't look like a chromosome. You need a sister. So, come here, I'll be my sister. Be my sister. Okay. We have to, like this. So you see? What are we connected with?
The arm.
This is not an arm fill. Centromere. Okay. So you will have to find a sister chromatid and to connect yourself with a centromere because we are not simulating interface. You need to find them now. We are quietly going to walk down the outside and we'll come back. Find yourself a sister.
Poncho.
Both is the best. Echo. The one has a hairy chest. I know. Silver too.
Where's some
I am definitely terrified.
if something half the size of a dog would kill me.
Alrighty. Yay.
That was fun.
I'm going to show you this on YouTube in a little bit. I got to get it uploaded. Alright, here's what I would like for you to do. Listen, I want to see your beautiful faces. What's your grade?
It's pretty.
I mean, it's okay. Some of us are not as pretty as others. Okay. I mean, oh, it's still videoing. Oh, I said that on. I have to edit that one out. Where are we at? Yep, it's still videoing.